As genetic science transforms our understanding of horse domestication, the implications extend far beyond academic knowledge to practical applications that will determine the future of human-horse relationships. The precision genetic tools emerging from domestication research promise to revolutionize how we breed, train, and care for horses, while simultaneously raising profound questions about the ethics of genetic modification. CRISPR gene editing represents the next frontier in this ongoing story. Researchers envision correcting genetic defects that cause equine diseases while potentially introducing beneficial genetic variants from some individuals into others. This could enable what scientists call precision breeding strategies achievable in one generation, rather than the decades typically required for traditional selection methods. However, most racing organizations currently prohibit genetically modified horses from competing, creating real tension between technological possibilities and sporting traditions. Personalized horse management based on genetic profiles is already emerging from research labs into practical application. Horses carrying PSSM1 mutations require very specific low-carbohydrate diets and particular exercise programs to stay healthy. Horses with certain MSTN genotypes benefit from different training approaches, sprint-oriented conditioning for some variants versus endurance-focused programs for others. Understanding these genetic predispositions enables targeted preventive care that can dramatically improve both horse welfare and performance outcomes. Conservation applications are becoming increasingly critical as we learn more about genetic diversity loss. Ancient DNA research has shown that modern horses represent only a limited fraction of the genetic diversity that was available to breeders in the past. Genetic tools now help identify the most genetically valuable breeding animals and design strategic outcrossing programs that maintain diversity while still preserving the characteristics that define each breed. The discoveries about ancient mutations are providing crucial insights for addressing modern veterinary problems. Understanding how the GSDMC gene strengthens horsebacks could potentially lead to new treatments for spinal dysfunction and back problems. Research on the DMRT3 neural circuits might inform therapies for various gait abnormalities. The basic science of domestication continues to yield unexpected practical applications for veterinary medicine. Economic pressures will almost certainly accelerate genetic technology adoption throughout the industry. With genetic testing becoming cheaper and more comprehensive each year, breeders who fail to adopt these tools risk serious competitive disadvantages. Industry projections suggest that genetic selection will become as routine in horse breeding as it already is in cattle and poultry production, a shift that's already well underway in many elite breeding programs. However, ethical frameworks are struggling to keep pace with technological capabilities. Scientists and ethicists are calling for clear guidelines that distinguish between therapeutic applications using genetics to treat diseases and prevent suffering and enhancement applications, improving performance beyond normal ranges. There's reasonable consensus supporting genetic testing for disease prevention, but enhancement applications remain deeply controversial and philosophically problematic. The story of genetic mutations making horses rideable ultimately demonstrates the profound interdependence between human and animal evolution. Just as humans developed horse rider syndrome skeletal modifications from intensive riding, horses underwent genetic changes that made them ideal human partners. This co-evolutionary relationship continues today as genetic science enables ever more precise partnerships between humans and horses. Perhaps most remarkably, the same mutations that enabled ancient horse domestication continue to influence every modern horse. Every smooth-gaited horse carries the ancient DMRT3 mutation that first appeared thousands of years ago. Every strong-backed riding horse benefits from GSDMC variants that were selected during the domestication bottleneck. Every gentle, trainable horse reflects ZFPM1 behavioral modifications that made the human-horse bond possible. The genetic legacy of those first rideable horses from 4,200 years ago lives on in stables worldwide, a testament to the enduring power of precise genetic changes to transform species relationships. As we stand at the threshold of an era when we can directly edit animal genomes with CRISPR precision, the story of natural genetic mutations enabling horse domestication provides both inspiration and cautionary guidance. The partnership between humans and horses, forged by ancient genetic accidents and refined by millennia of selection, remains one of biology's most successful collaborations, offering profound lessons for creating ethical and beneficial relationships with the animals that share our world. Think about what this means for your own relationship with horses if you're a rider or owner. Every time you saddle up, you're benefiting from genetic changes that occurred millennia ago. The comfortable ride you experience, the willingness of your horse to accept your cues, even the smooth gait that keeps you balanced. All of these trace back to specific mutations that were preserved because they made human-horse partnership more effective. The research continues to evolve rapidly. Scientists are discovering new genes involved in domestication almost every year, adding layers of complexity to what we thought was already a complete picture. Recent studies have identified additional behavioral genes, variants affecting disease resistance, and modifications that influence everything from coat color to metabolic efficiency. Each discovery adds another piece to the puzzle of how wild animals became our closest working partners. For modern horse owners and breeders, these insights create both opportunities and responsibilities. Understanding the genetic basis of traits allows us to make better breeding decisions, but it also requires us to think carefully about what we're selecting for and why. Are we prioritizing performance at the expense of health? Are we maintaining enough genetic diversity to ensure long-term population viability? These aren't just academic questions. They determine the future welfare of horses worldwide. The ethical dimension becomes even more pressing as technology advances. If we can use CRISPR to eliminate genetic diseases, should we? Most people would probably say yes. But what about using it to enhance speed, jumping ability, or endurance beyond natural ranges? Where do we draw the line between therapeutic intervention and performance enhancement? These questions don't have easy answers, but the genetic research on domestication gives us a framework for thinking about them. One fascinating aspect that often gets overlooked is how this research changes our understanding of agency in domestication. For years, we talked about domestication as something humans did to animals. We tamed them, we bred them, we shaped them to our needs. But the genetic evidence suggests a more nuanced story. Those mutations arose spontaneously, and horses carrying them thrived because they were better suited to the new ecological niche created by human partnership. In a sense, horses with the right mutations domesticated themselves by succeeding in a human-modified environment. This perspective doesn't diminish human influence, but it recognizes that domestication was truly a partnership from the beginning. Horses weren't passive recipients of human selection. They were active participants in a co-evolutionary process that changed both species. The humans who learned to ride developed modified skeletons. The horses who could be ridden developed modified genetics. Both species adapted to each other. The speed at which the GSDMC mutation spread, 10 to 20 times faster than human lactose tolerance, tells us something important about selection pressure. This wasn't gradual change over countless generations. Once rideable horses appeared, they spread explosively because they provided such enormous advantages. Societies with rideable horses could travel farther, fight more effectively, and trade more extensively. The mutation succeeded because it opened up entirely new possibilities for both humans and horses. Looking forward, we're entering an era where we can potentially guide these evolutionary processes with unprecedented precision. Genetic testing already allows breeders to identify carriers of disease-causing mutations and avoid producing affected offspring. Gene editing could theoretically eliminate these harmful variants entirely within a few generations. But with this power comes responsibility to consider long-term consequences and maintain the genetic diversity that allows horse populations to adapt to future challenges. The story of horse domestication ultimately teaches us humility. For all our intelligence and technology, the horses we ride today depend on random mutations that occurred thousands of years ago. We didn't engineer DMRT3 or GSDMC. We simply recognized and preserved horses that carried these variants because they served our purposes. Nature provided the innovations, humans provided the selection pressure that made them succeed. As research continues and technology advances, we have opportunities our ancestors could never have imagined. We can read the complete genetic code of any horse, identify the variants that affect every trait, and make breeding decisions with extraordinary precision. We're even approaching the ability to write new genetic instructions, creating modifications that nature never produced on its own. The question isn't whether we can do these things, it's whether we should and how we can do so responsibly. Have you ever wondered why horses let us ride them? Why they're so uniquely suited to carrying humans on their backs for hours without complaint? The answer might surprise you. It turns out that riding horses wasn't always possible. In fact, recent scientific breakthroughs suggest that a single genetic mutation occurring around 4,200 years ago literally transformed horse spines and made comfortable riding feasible for the first time in history. This isn't some minor evolutionary tweak we're talking about. Research published in 2024 identified a specific gene called GSDMC that underwent intense selection pressure during horse domestication. This mutation spread through horse populations at an astonishing rate, somewhere between 10 to 20 times faster than human lactose tolerance evolved. Why so fast? Because horses with this mutation could comfortably carry riders across vast distances, giving them an enormous survival advantage in a world where humans were becoming the dominant force. Here's where it gets really interesting. The same GSDMC gene variant that made horses perfect for riding actually causes chronic back pain in humans. But in horses, this genetic change strengthened their vertebrae, enhanced their motor coordination, and created the sturdy foundation needed to support a human rider. Without this precise modification, along with another famous mutation in the DMRT3 gene known as the gatekeeper, horses might have remained animals we used primarily for milk and meat, rather than transportation and partnership. Let's talk about one of the most groundbreaking discoveries in horse genetics. Back in 2012, Dr. Leif Andersson's team at Uppsala University identified something remarkable, a mutation in the DMRT3 gene that they nicknamed the gatekeeper. This wasn't your typical genetic variant. It represented a completely new way of controlling movement in vertebrates that scientists had never observed before. The mutation does something fascinating at the molecular level. It creates what's called a premature stop codon at position 301 on the gene, which produces a shortened, truncated protein. This altered protein acts as what scientists call a dominant negative form, meaning it actively interferes with the normal function. But here's the crucial part. This genetic change specifically rewires the neural networks in the spinal cord. In a normal horse, specific neurons in the spinal cord ensure strict diagonal gait patterns. When the right front leg moves forward, the left hind leg moves at exactly the same time. This diagonal coordination is hardwired into their nervous system, but the DMRT3 mutation disrupts this rigid pattern, allowing horses to develop entirely new ways of moving. The practical results are extraordinary. Horses carrying this mutation can perform what are called lateral gaits movements, like pacing and tolt, where same-side legs move together instead of diagonal pairs. If you've ever ridden a Paso Fino or an Icelandic horse, you've experienced this firsthand. These gaits are so smooth that riders often say you could carry a glass of wine without spilling a drop. By 2012, the mutation had reached 94% frequency in Icelandic horses. Clear evidence of intense selection pressure favoring these comfortable gaits. But the DMRT3 gene is only part of the story. The 2024 research by Ludovic Orlando's team uncovered an even more fundamental piece of the puzzle, the GSDMC gene mutation. This discovery, published in the journal Science, traced the mutation's emergence to about 4,750 years ago, right during the critical domestication bottleneck when horses were transitioning from wild to domestic animals. What makes this finding so remarkable is what the gene actually does. In humans, GSDMC variants are associated with chronic back pain and serious spinal problems. Yet in horses, this same genetic change created stronger vertebrae, better motor coordination, and improved muscular strength throughout the back and trunk. Scientists tested this by studying mice with altered GSDMC genes and found the animals developed straighter spines, stronger limbs, and significantly better locomotion. Exactly the changes needed to transform a horse into an effective riding animal. There's a third critical mutation that often gets overlooked because it affects behavior rather than physical structure. The ZFPM1 gene underwent selection around 5,000 years ago and plays a crucial role in how the brain develops, specifically affecting serotonergic neurons. These neurons are central to regulating mood, anxiety, and stress responses. The mutation in ZFPM1 essentially dialed down the horse is natural fear and aggression while increasing docility and stress tolerance. Think about what this means practically. Without this psychological adaptation, horses would have remained too fearful and reactive to work alongside humans, no matter how perfect their bodies were for riding. A horse with a strong back and smooth gaits is useless as a riding partner if it panics every time you approach it. The story of when and where horses were actually domesticated has undergone a complete revolution thanks to ancient DNA analysis. For decades, scientists were convinced they had it figured out. The Botai culture in Kazakhstan, dating back 5,500 years, seemed like the obvious answer. They had horse corrals, their horse teeth showed what looked like bitware, and archaeological evidence suggested intensive horse management. Case closed, right? Wrong. When researchers analyzed DNA from 273 ancient horse skeletons in 2021, they discovered something shocking. Those Botai horses weren't the ancestors of modern domestic horses at all. They were actually the ancestors of Przewalski's horses, the wild horses that still exist today in Mongolia and parts of China. So, when did domestication really happen? The genetic evidence points to a completely different time and place. The Western Eurasian steppes, specifically the lower Volga-Don region, around 4,200 years ago. These horses, which scientists call the Dom-2 lineage, carry the genetic signatures that show up in every single modern domestic horse breed, and once these horses appeared, they spread across Eurasia with breathtaking speed. By 2000 BCE, they had reached from the Atlantic Ocean all the way to the Pacific, completely replacing local horse populations in what might be the fastest large mammal expansion in prehistory. This timeline tells us something crucial about those genetic mutations we discussed earlier. The GSDMC spine-strengthening mutation began its rapid spread precisely during this domestication bottleneck. This timing suggests that physical radiability was the key innovation, the secret sauce that made these particular horses so incredibly successful. Soon after domestication, archaeological evidence shows horses being used for warfare and long-distance travel, coinciding perfectly with the emergence of chariot technology and the expansion of Indo-Iranian cultures across the continent. Looking at Przewalski's horses today really drives home how profound these genetic changes were. These wild horses have 66 chromosomes compared to 64 in domestic horses. They're significantly smaller too, weighing between 550 and 800 pounds, while domestic horses range from 400 to well over 1,000 pounds. But the differences go far beyond size and chromosome numbers. Przewalski's horses are highly aggressive and maintain powerful flight responses. They show very limited capacity for bonding with humans and remain essentially untrainable for riding purposes. Most importantly for our story, they completely lack the DMRT3 and GSDMC mutations that made smooth, comfortable riding possible. Trying to ride a Przewalski's horse would be uncomfortable at best and dangerous at worst. They simply weren't built for it, either physically or psychologically. The coevolution worked both ways too. While horses were developing genetic changes that enabled rideability, humans were developing what anthropologists call horse rider syndrome, distinctive skeletal modifications visible in human remains from 5,000 years ago. These permanent changes to human bone structure prove that mounted riding was intensive enough to physically alter our own anatomy. It's a fascinating example of how deeply intertwined human and horse evolution became. To really understand how these genetic mutations translated into rideable horses, we need to look at the sophisticated biomechanical systems that enable comfortable human transport. Think of the horse's back as functioning like a suspension bridge between the fore and hindquarters. The primary support system for this bridge is the longissimus dorsi muscle, an absolutely massive muscle that runs along the spine, measuring over 2 meters long and weighing up to 5 kilograms in a large horse. The GSDMC gene mutation specifically enhanced this entire system by strengthening the vertebral structure and improving muscle coordination throughout the back region. A horse has 7 cervical vertebrae in the neck, 18 thoracic vertebrae in the back where the saddle sits, and 6 lumbar vertebrae in the loin area. The thoracic region forms a rigid platform because the ribs attach to these vertebrae, but it takes more than just bones to create an effective saddle platform. The GSDMC mutation enhanced the ligament systems and muscle development that transform this skeletal framework into something that can comfortably support a rider for hours. The DMRT3 mutation revolutionized how horses move by altering what scientists call central pattern generators, or CPGs, in the spinal cord. These are neural networks that control rhythmic limb movements. Normal horses are essentially locked into diagonal coordination patterns, but the mutation enables lateral gaits by disrupting specific inhibitory interneuron networks. This creates the smooth, 4-beat tilt gait that provides minimal vertical displacement for riders. In other words, less bouncing. High-speed video analysis has revealed the biomechanical advantages in precise detail. Alternative gaits reduce rider bouncing by up to 70% compared to trotting, while also being more energy efficient for horses at intermediate speeds. The mutation enables horses to maintain these comfortable gaits consistently, which was almost certainly a crucial advantage for long-distance travel and likely drove their rapid adoption across Eurasia. The anatomical changes extended well beyond just the spine. Domestic horses evolved larger overall body size, longer legs relative to body size, and enhanced muscle development patterns specifically adapted for carrying loads. When you compare modern horses to Przewalski's horses, you see increased back length, more refined head and neck proportions, and specialized muscle fiber arrangements in the longissimus dorsi that optimize rider support. Modern biomechanical studies using electromyography and force plate analysis have revealed even more about the sophisticated neuromuscular control systems that enable riding. The DMRT3 mutation affects not just the pattern of movement, but also the precise timing and coordination of muscle activation. This creates the predictable, balanced movement that makes horses safe and comfortable for human riders, something that can't be trained into a horse that lacks the genetic foundation. The discoveries about these ancient genetic mutations have completely transformed how we breed horses today. Commercial genetic testing now allows breeders to predict offspring traits with a level of accuracy that would have seemed like science fiction just a generation ago. We can now analyze everything from coat color to performance capabilities to disease susceptibilities before a foal is even conceived. Companies like Equine genetics offer comprehensive test panels that examine more than 75 different genetic traits. UC Davis Veterinary Genetics Laboratory provides breed-specific health testing that can identify carriers of genetic diseases. The American Quarter Horse Association has even made genetic testing mandatory for breeding stallions, requiring screening for 5 major genetic diseases that can cause serious suffering in affected foals. This represents a fundamental shift from traditional selection based on appearance and performance to precision genetic management. The DMRT3 gait gene has become a particularly powerful tool for breeders. Horses that carry 2 copies of the mutation, what geneticists call homozygous AA genotype, show this variant in nearly 100% of gaited breeds, like Tennessee walking horses and standard bred pacers. Meanwhile, sport horses bred for jumping and dressage typically have the wild-type CC genotype that enables the diagonal gait superior for those disciplines. Breeders can now definitively predict gait characteristics before foals are even born. Recent research has identified additional genes that modify gaits beyond just DMRT3. The RELN gene affects both gait quality and physical conformation, while STEU2 influences motor learning ability, essentially how quickly and well a horse can learn new movements. When you combine the effects of DMRT3 with these other variants, scientists can now explain about 27% of the variation in pacing ability across different horses. This enables increasingly precise breeding decisions. The cutting edge of research is pushing into even more controversial territory with genetic modification. Argentine scientists have successfully created CRISPR-edited horse embryos targeting the myostatin gene, often called the speed gene, to enhance muscle development. While such horses cannot currently compete due to racing regulations that prohibit genetic modification, the technology clearly demonstrates what's possible in the near future. The economic impact of genetic testing has been substantial. It has become standard practice in elite breeding operations, with horses commanding premium prices based on their genetic profiles. Industry analysis shows that genetic tools reduce financial losses from inherited diseases while accelerating genetic progress by enabling more informed breeding decisions. However, there's a concerning trend that researchers are tracking closely. Modern horses have lost about 16% of their genetic diversity in just the past 200 years as breed development has intensified. Scientists worry that our intensive selection for specific performance traits may be compromising overall genetic health. We might be accumulating deleterious mutations, harmful genetic variants, while simultaneously reducing the gene pool's resilience and ability to adapt to changing conditions. Despite all these revolutionary genetic discoveries, fierce scientific controversies continue to rage about how horses actually became rideable. The neat narrative of genetic mutations enabling domestication faces significant challenges from researchers who argue for alternative explanations. The most contentious debate still surrounds the famous Botai culture. While most scientists now accept that genetic evidence disproves Botai domestication, William Taylor and Christian Baron Ortiz published a 2021 study that stirred up serious controversy. They argued that what everyone had been calling bit wear on Botai horse teeth actually represents natural dental development issues and pathologies. They contend that Botai represents intensive wild horse hunting rather than true domestication, comparing the wear patterns to those found in completely wild Pleistocene horses from North America that lived thousands of years before humans even thought about riding. This sparked furious rebuttals from Alan Outram and other researchers who accused Taylor's team of fundamental scientific misunderstanding and misrepresenting key methodologies. The battle continues to rage because the same archaeological evidence apparently supports completely different interpretations, keeping the scientific community deeply divided about what early horse management actually looked like. Some critics question whether genetic mutations alone can really explain the domestication process. Even Ludovic Orlando himself, one of the lead researchers who discovered the GSDMC mutation, acknowledges that complex traits are only exceptionally determined by single genes, and that most traits involve small contributions of a large number of genetic variants. The focus on GSDMC and DMRT3 genes might be oversimplifying an incredibly complex process that actually involved hundreds or even thousands of genetic changes working together. Alternative theories propose completely different drivers of domestication. Some researchers argue that late Neolithic climate cooling boosted wild horse populations and expanded their range, making them more accessible to humans without requiring any genetic modifications at all. Others suggest that improved handling techniques, better corral designs, and the invention of halters and other equipment were more important than biological changes in the horses themselves. Ethical concerns complicate the scientific picture even further. The discovery that beneficial mutations, like GSDMC, actually cause chronic back pain in humans raises disturbing questions. Are we celebrating genetic changes that created long-term suffering in horses? Some scientists argue that reducing natural fear responses through the ZFPM1 mutation might compromise horses' ability to protect themselves from danger, representing a form of psychological suppression rather than genuine behavioral improvement. These modern breeding controversies extend directly from these debates. While genetic testing can prevent inherited diseases, critics worry about commodifying animals based on genetic profiles and prioritizing performance over actual welfare. The revelation that modern horses carry accumulated harmful mutations suggests that centuries of artificial selection have created ongoing health problems, even while enhancing the specific traits we value for competition and work. As genetic science transforms our understanding of horse domestication, the implications extend far beyond academic knowledge to practical applications that will shape the future of human-horse relationships. The precision genetic tools emerging from domestication research promise to revolutionize how we breed, train, and care for horses, while raising profound questions about the ethics of genetic modification. CRISPR gene editing represents the next frontier. Researchers envision correcting genetic defects that cause equine diseases while potentially introducing beneficial alleles from some individuals into others. This could enable precision breeding strategies achievable in one generation, rather than the decades typically required for traditional selection. However, most racing organizations prohibit genetically modified horses, creating tension between technological possibilities and sporting traditions. Personalized horse management based on genetic profiles is already emerging. Horses with PSSM1 mutations require specific low-carbohydrate diets and exercise programs, while MSTN genotyping helps trainers tailor conditioning for sprint versus endurance disciplines. Understanding genetic predispositions enables targeted preventive care that can dramatically improve horse welfare and performance. Conservation applications are increasingly critical. Ancient DNA research reveals that modern horses represent only a limited fraction of the genetic diversity available to past breeders. Genetic tools now help identify the most genetically valuable breeding animals and design outcrossing programs to maintain diversity while preserving breed characteristics. The discoveries about ancient mutations provide crucial insights for addressing modern problems. Understanding how the GSDMC gene strengthens horsebacks could lead to new treatments for spinal dysfunction, while research on DMRT3 neural circuits might inform therapies for gait abnormalities. The basic science of domestication continues to yield practical applications for veterinary medicine. Economic pressures will likely accelerate genetic adoption. With genetic testing becoming cheaper and more comprehensive, breeders who fail to adopt these tools risk competitive disadvantages. Industry projections suggest that genetic selection will become as routine in horse breeding as it already is in cattle and poultry production. However, ethical frameworks lag behind technological capabilities. Scientists call for clear guidelines distinguishing between therapeutic applications, treating diseases, and enhancement applications, improving performance beyond normal ranges. The consensus supports genetic testing for disease prevention, but enhancement applications remain controversial. The story of genetic mutations making horses rideable ultimately reveals the profound interdependence between human and animal evolution. Just as humans developed horse rider syndrome, skeletal modifications from intensive riding, horses underwent genetic changes that made them ideal human partners. This co-evolutionary relationship continues today as genetic science enables ever more precise partnerships between humans and horses. Perhaps most remarkably, the same mutations that enabled ancient horse domestication continue to influence modern horses. Every smooth-gaited horse carries the ancient DMRT3 mutation, every strong-backed riding horse benefits from GSDMC variants, and every gentle, trainable horse reflects ZFPM1 behavioral modifications. The genetic legacy of those first rideable horses 4,200 years ago lives on in stables worldwide, a testament to the enduring power of precise genetic changes to transform species relationships. As we stand at the threshold of an era when we can directly edit animal genomes with CRISPR precision, the story of natural genetic mutations enabling horse domestication provides both inspiration and cautionary guidance. The partnership between humans and horses, forged by ancient genetic accidents and refined by millennia of selection, remains one of biology's most successful collaborations, offering lessons for creating ethical, beneficial relationships with the animals that share our world. So the next time you're around horses, take a moment to appreciate the deep history encoded in their DNA. Those smooth gaits, that willingness to carry a rider, that calm temperament in the face of strange situations. All of these trace back to specific genetic changes that made the human-horse partnership possible. We're not just working with horses, we're participants in a 4,200-year-old co-evolutionary relationship that continues to unfold today, shaped increasingly by our growing understanding of the genetic foundation that makes it all possible. The genetic revolution that made horses rideable didn't end in ancient times. It continues in research laboratories, veterinary clinics, and breeding barns around the world. Every genetic test, every breeding decision, every advance in our understanding of equine genetics adds another chapter to this extraordinary story. We are privileged to live in an era when we can finally read the genetic text that explains how one of history's most important partnerships came to be, and perhaps use that knowledge to ensure it continues successfully for generations to come. Thanks so much for watching this exploration of how genetic mutations made horses rideable. If you found this fascinating, please hit that like button. It really helps the channel grow and allows me to create more content like this. And definitely subscribe if you want to see more videos exploring the science behind the animals we work with every day. There's so much more to discover about the genetic stories written in the DNA of every creature around us. Until next time, Take care, and maybe give your horse an extra pat. They're carrying some pretty remarkable genetic history on their backs.